This is week 3 of biology, the first quarter, and today we're going to talk about the transportation of substances, the molecular membrane, and lastly we will talk about the formation of energy within the cell and the chemical reactions involved in the formation of energy. To start out, we have to discuss in detail, a modest amount of detail, the plasma membrane. Now, the plasma membrane is called a phospholipid bilayer. So let's take that or unpack that word phospholipid and bilayer apart. This is what's known as the phospholipid, and this is a expanded picture of a phospholipid. A phospholipid is a situation where the lipid, one of the fatty acid molecules, has been replaced by a phosphate group. This is the phosphate group up here. And so what you have is glycerol, which is this molecule right here. If you had OHs, it would be glycerol. Then attached to that are fatty acids. And this is the acid part right here. And normally there would be a third one, which is why fats are called triglycerides. They're the combination of glycerol and 3 fatty acids, 3 being tri, so triglyceride. Well, in this case, we have a phosphate substituted, so this is what's known as a phospholipid. This is then arranged with the heads of the phosphate groups together outside of the cell and inside of the cell, and the tails, or the lipid part, facing each other in the middle. Thus the bilayer, okay? And this forms a hydrophilic end, which is the water-loving, philic meaning loving. This is the polar part of the molecule. This is polar and is charged. And if you remember your basic science, water is a polar solvent. Therefore, like dissolves like, so water is loving or can associate itself with this part of the molecule. Now this part of the molecule is called hydrophilic, and hydrophilic means that it's water-fearing, hydrophobic, water-fearing. It's the non-polar aspect of the molecule, and therefore water does not mix in this part of the lipid bilayer. And it's just like if you put water and oil in a glass and shook it up, it would separate because they do not mix together. So you have water soluble and water soluble on both sides, but within the middle is water insoluble. And this presents a fairly impregnable barrier to water. Now this is a more complex view of the same situation. You have a phospholipid bilayer in which the phospholipids are arranged in 2 layers, but also in the membrane are several other structures, glycoprotein structures and glycolipid structures. And these, glycoprotein is just a structure where a carbohydrate is attached to a protein. A glycolipid is just where a carbohydrate is attached to a lipid. But you can see that these can, in many ways, allow for the passage of molecules that would not normally pass through this membrane. So with that introduction to the membrane structure, let's take a look at some ways in which the molecules can pass through. First of all, we have what's known as passive transport. Now, passive transport is the movement of molecules across the lipid bilayer according to the laws of diffusion and osmosis. This means that the molecules obey the basic laws of physics regarding diffusion. That is, that higher concentration, the molecules will move away from higher concentrations and into lower concentrations. The other form of transportation is active transportation, and this is the movement of molecules through the plasma membrane aided by energy expenditure. Now, commonly, the reason that this has to be done is because it's actually against the laws of diffusion and osmosis. And so the cell will actively pump molecules either inside the cell or outside the cell, and the energy is required, and if this energy was not expended, then the molecules wouldn't be there. This then sets up a situation where you have three possible concentration gradients, if you will, between the inside of the cell and the outside of the cell. And you can talk about this in terms of the solution outside the cell. Now, if the solution inside the cell and outside the cell has the same number of molecules or the same concentration of molecules, it's called isotonic. And this is shown in the middle of the slide. The concentrations is essentially equal. Now, it does not mean that those same molecules stay on the outside and the same molecules stay on the inside. Molecules pass in and out of the cell, but the concentration never changes. And this is known as a dynamic equilibrium because things are moving, but the overall situation does not change. This is like people walking inside and outside the room at the same rate, such that the number of people in the room always remain the same. The second possible situation is shown on the right, and this is where the number or concentration of molecules outside of the cell is much higher than the concentration within the cell. And this is known as a hypertonic solution. Now, the laws of diffusion would say that the fluid would move from within the cell out of the cell to decrease the concentration outside the cell, or that molecules would move from outside the cell inside the cell to achieve the same effect. The third situation is shown on the left. In this case, the concentration outside of the cell is much lower than the concentration inside the cell, and this is known as a hypotonic solution. In this case, the natural inclination of fluid is to move inside the cell from outside the cell, or for the molecules to move from inside the cell outside the cell. So, these are the three possible situations that can occur. Now, if fluid is allowed to flow freely across the plasma membrane, these various types of solutions can result in three different states of the cell. The first state is shown on the left in a hypertonic situation, and in this case, the term that's used is shrunk because fluid is passed from within the cell to outside the cell, and the cell itself collapses. This is also called plasmolysis. It's the collapse of the walled cell cytoplasm due to a lack of water, because the water is all passed out of the cell in order to try to dilute the concentration of the molecules outside the cell. The center is the normal situation, and on the right is a situation where you have low concentration outside the cell, so fluid flows from outside to inside the cell. The cell becomes swollen and then ultimately can rupture. And when it ruptures, it's called cytolysis. It's the rupturing of the cell wall due to an excessive internal fluid pressure that's caused by the passage of fluid passively into the cell from outside the cell, trying to raise the concentration of molecules outside the cell and lower the concentration of molecules inside the cell. So these are the three things that can occur, and they do occur in the blood system from time to time. Now, in terms of energy production, there are several ways in which energy is produced. And the diagram shows the normal energy production. There are actually 4 steps in normal energy production within the cell. And 3 of the 4 steps are pictured here, the 3 that actually produce energy. That is what is called cellular respiration. It's the processes of chemical reactions that result in the production of energy. And glycolysis, the Krebs cycle, and electron transport chain are the 3 energy producing steps of cellular respiration. I've also included the definition of activation energy here because a certain amount of energy is actually required for any chemical reaction to actually start and move forward. So as I said, glycolysis is the first step in the production of energy. The next step in the production of energy is called the Krebs cycle. It's also known as the citric acid cycle. This is the third step. I call it the next step in fact that it's the next step that produces energy. In this process, 6 glucose molecules, once converted to pyruvate, are changed to 6 carbon dioxide molecules, 12 hydrogen atoms, and 3 oxygen molecules are consumed. And I'm going to show you the chemical reactions that occur that result in this outcome. The last process is called the electron transport system, or the electron transport chain. This by far produces the most energy of the 3 energy producing processes, and in this process hydrogen is reacted with oxygen to make water. And as I point out here, this is exactly the same process that occurs in a hydrogen fuel cell. Here's the first equation for glycolysis. And what you see here is the chemical formula for glucose, C6H12O6. That just means that there are 6 carbons, 12 hydrogens, and 6 oxygens in glucose. This is broken in half to form pyruvate, which is C3H4O3, and 4 hydrogen atoms. And if you count up all the atoms on both sides of the equation, you'll see that they're the same number on the left side of the equation as on the right side. And this produces energy. The next step, which actually does not produce energy, although I've put the word energy in here, is called the formation of acetyl-CoA. In this step, 2 pyruvate molecules are changed into 2 acetyl-CoA molecules plus carbon dioxide plus hydrogen atoms. The acetyl-CoA molecules are the 2 molecules that actually enter the Krebs cycle. Once they enter the Krebs cycle, 3 oxygen molecules are combined with them, and 4 carbon dioxide and 6 hydrogen atoms are produced. And again, more energy is produced. And lastly, in the electron transport system, 12 hydrogen atoms are combined with 3 oxygen molecules to form 6 molecules of water. So you can see that overall, glucose is broken down to form 6 carbon dioxides and 6 water molecules, and energy is produced. Now, I've shown in this diagram ATP, and that is the form in which this energy takes, and I want to spend just a minute to talk about that. This is the structure of ATP. You have an adenosine, a ribose sugar, and 3 phosphates. So that's adenosine triphosphate is the name of the molecule. And this is the molecule in which all the released energy from cellular respiration is stored. So think of it this way. Let's say I go out and cut grass, and I need something to store that effort that I put into that. And so somebody gives me $5. So that $5 is the same as this ATP. Then I can take that $5 and give it to somebody else, and they will give me a very nice banana split. So when you break down the ATP to form ADP, energy is released, and that's like spending your money. And energy is released, and it powers whatever process you want to do. So in a way, I've called ATP the energy currency in the cell. It's the currency in which the cell uses to produce, and when it produces energy, it converts it into this currency, and then when it spends energy, it breaks it off of this currency. So let's look at the way in which we produce energy, or how much energy is produced. So first we have glycolysis. Again, it's the fracturing of a glucose molecule into 2 pyruvate molecules and 4 hydrogen atoms. And generally it's accepted that 2 ATPs are formed when this occurs. The next step, which is shown here, is the formation of acetyl-CoA. And as I said, there are actually no ATP forms with acetyl-CoA. And you can see in the diagram, it says 0. And sometimes they say you can get a little bit of energy from this. And again, it says depending on shuttle that transports electrons from ADH. That you don't need to think about. But generally, it's accepted that no energy is formed with the formation of acetyl-CoA. Then the Krebs cycle turns. And in the process of making these conversions, another 2 ATPs are formed. And then lastly, we go through our fuel cell reaction, which is 12 hydrogens plus 3 oxygens form 6 water molecules. And in that process, anywhere from 32 to 34 ATPs are formed. So in the entire process, a total of 36 ATPs are formed. With each molecule of glucose results in 36 ATPs of 36 ATPs, with the consumption of 6 molecules of oxygen, which I haven't shown here, and the production of 6 molecules of carbon dioxide and 6 molecules of water. Now you know we have to get rid of this carbon dioxide, and that occurs when we breathe out. And we have to get the oxygen back, and that occurs when we breathe in. And the water is taken care of in our water balance when we go to the bathroom and drink water. And so all of this goes along and produces a substantial amount of energy for each molecule of oxygen of glucose that's burned. Now the last thing I want to talk about, wrapping this up, and we're only going to have one part to this lecture because it's a very short lecture, only about 20 minutes long, is what occurs in a less than adequate oxygen environment. Everything that I've shown you up to this point requires sufficient oxygen in order for it to occur. In both animal and plant systems, there are many situations where adequate oxygen is not present, and some systems in which oxygen is preferred. And one process that occurs when inadequate oxygen is present is called cellular fermentation. Cellular fermentation is the conversion of either of pyruvate molecules to lactic acid or to alcohol. And this depends on the system that you're in. In yeast systems, for example, the conversion is from glucose to pyruvate to alcohol. In human muscle, the conversion is from glucose to pyruvate to lactic acid. And both of these occur in an environment in which there's not enough oxygen for a normal process to occur. Here's an example of when alcohol is formed, and this is what occurs in yeast cells. What you have is glucose being converted to pyruvate, just like the glycolysis that we've seen before. But instead of being then converted to acetyl-CoA, 2 carbon dioxide molecules are stripped off and you form something called acetaldehyde. This is then converted from acetaldehyde to alcohol. And this is the process that happens when you make beer or wine. You start out with glucose, you have organisms that are present, yeast organisms or other types of organisms, and they convert with the production of carbon dioxide, they convert that glucose into alcohol. And this is what is known as fermentation. Now the other process that occurs in animal systems is called anaerobic metabolism or lactic acidosis. And this is what occurs in that situation. In a situation where there is no oxygen present, the glucose is converted directly into lactic acid. In that situation, ATP is produced, but it is a far less ATP than you would produce if you had adequate oxygen present. And on top of that, so this is an emergency situation that allows for the production of energy, but has the downside of being far less efficient in terms of energy production. And also you produce lactic acid. Lactic acid is bad. and causes something known as lactic acidosis. This is the final result of cellular fermentation that occurs in muscle and other mammalian situations. And the lactic acidosis is not good for human systems and tends to cause serious problems when it occurs. So this is the end of our lecture today. It was only 20 minutes long. I guarantee you that you'll appreciate these shorter lectures because when we get into some other things, the lectures will be longer and have more parts. So take advantage of these shorter lectures at this time. And I will see you in class on Monday.